Hello, how are you? Hi. Hi. Yeah. All right. Welcome. Uh, so Varshith, did you want to do a presentation today or? Okay. That sounds good. Yeah. Thank you. Okay. That sounds good. Thank you. Uh, oh yeah. Yeah. All right. That sounds good. All right. Thank you, Varshith. Susan, did you have anything to update us on? All right. Yeah. Yeah, I can't, but well, that's great. Yeah, you should put. Yeah. Yes. Okay. So these questions, how do you envision them? Like, so let's go through them. Yeah. So this is, does the embryo's future depend only on its current shape, or does it remember the past? I don't think it's about remembering the past. Yeah. Okay. Well, I mean, that's you would need more than just kind of like the lineage tree data for that. Probably like, uh, yeah. But it's yeah. I mean, C. elegans is known for being kind of a deterministic model of cell division. In other words, we know what the cells will, what the outcomes will be. The only time that's really in question is if you're dealing with a mutant or something like that. So there may be ways to use that method to predict like mutant phenotypes and things like that, which would involve like, just setting up sort of almost like a simulated data set where you kind of leave cells out and then you say, well, I'll leave this cell out or this part of the cell lineage out. What does the shape look like given that it's like missing? I get maybe that's one way to do it. I'm trying to think of other ways to do this, but um, being able to take like information from the past and fill in, you know, uh, when you have something missing, basically what gets filled in there, what gets substituted, what gets compensated. Yeah. All right. Yeah. I mean, yeah. So we could do that. I mean, you'd have to think through the problem a little bit, but to like kind of maybe generate a altered data set based on like kind of what it's going to pick up on or something. Um, and you can also merge some of these lineage tree data sets with molecular data or genetic data, so we know kind of what the major phenotypes are or what the major mutations are that are linked to major phenotypes in the alternate phenotypes in the embryo. So we can look at that as well. Um, So I don't know. We'll have to think about that some more. Yeah, and then the second question is, can we make a probability of the embryo moving from one state to the other? So that's kind of like a Markovian type thing. Yeah, which is, it's again, you could probably do that pretty easily in C. elegans. You're talking about the entire embryo just kind of shifting from one state to another, like you were showing the gastrulation state, or I don't remember that slide. Yeah, 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 that one. Yeah, so that might be interesting. Yeah, and then number three is make it easy for yeah. All right. Yeah. yeah, that would be good. I mean, we've kind of done that in the past, but not really that much. Right. Yeah. Well, yeah, I think that's yeah, I think three would be where that question would land. Like we were doing things topological data analysis, but there are other ways to visualize things. So all right, thank you. Very good. There any other questions for people have for Barshan? All right, that sounds good. So yeah, well, go ahead. Yeah. So Barshan also posted on this Nature paper. This is right here, an end-to-end attention-based approach for learning on graphs. These are the authors here. This is in Nature Communications. So their interest is in thinking about the transformer-based attention approach that we use for machine learning, for deep learning, and then they're going to add this new approach to it. So there has been a recent surge in transformer-based architectures for learning on graphs, mainly motivated by attention as an effective learning mechanism and the desire to supersede the handcrafted operators characteristic of message passing schemes. So we use message passing to represent information flow in the network or changes in the edges of the network. However, concerns over their empirical effectiveness, scalability, and complexity of the preprocessing steps have been raised, especially in relation to much simpler graph neural networks that typically perform on par with them across a wide range of benchmarks. To address these shortcomings, we consider graphs as edges or sets of edges and propose a purely attention based approach consisting of an encoder and an attention pooling mechanism. The encoder vertically interleaves masked and vanilla self attention modules to learn an effective representation of edges while allowing for tackling possible misspecifications in input graphs. Despite its simplicity, the approach outperforms fine-tuned message passing baselines and recently proposed transformer-based methods on more than 70 node and graph level tasks, including challenging long-range benchmarks. Moreover, we demonstrate state-of-the-art performance across different tasks, ranging from molecular to vision graphs and heterophilous node classification. The approach outperforms graph neural network and transformers in transfer learning settings and scales much better than alternatives with a similar performance level or expressive power. So this goes into what they're doing here with this model. They're kind of building on this purely attention-based approach. So you basically, this is in graph neural networks, which is a little bit different than the way that these things are done in regular neural networks. So they talk about how to learn from graphs. The resulting graph neural network typically stack multiple such layers, which are these basic layers of neural network, to learn node representations and vertex-rooted subtrees, essentially mimicking the one-dimensional Weisfeiler-Lehmann or 1WL graph isomorphism test, which is that they use oftentimes in the field to measure things like graph isomorphism, so which graph topologies are equivalent, things like that. Variations of message passing have been applied effectively in different fields such as life sciences, electrical engineering, and weather prediction. So they talk about several practical challenges that have presented themselves. Although the message passing framework is highly flexible, the design of new layers is a challenging research problem. This is something that people generally use handcrafted operators for. And so, it's a little bit constraining because you have to kind of deal with dynamic data and things like that. So the nature of message passing also imposes certain limitations that have shaped the majority of the literature. One of the most prominent examples is the readout function used to combine node-level features into a single graph-level representation, which is required to be permutation invariant with respect to node order. So there are a lot of things with node order and isomorphisms that you have to kind of overcome that are kind of still challenges to building these kind of graphs from data. And so they kind of go through some of the challenges here. Alternative solutions for these two problems typically take the form of message regularization schemes. There is, however, no clear consensus on the right architectural choices for building effective deep message passing neural networks and effectively addressing all these challenges. Transfer learning and strategies such as pre-training and fine-tuning are also yet less ubiquitous in graph neural networks because of modest or ambiguous benefits, as opposed to what we see in large language models. So they're focusing on the attention mechanism, and they talk about kind of how these can be used in GNNs. And then, motivated by the effectiveness of attention as a learning mechanism and recent advances in efficient and exact attention, we introduce an end-to-end attention-based architecture for learning on graphs that is simple to implement, scalable, and achieve state-of-the-art results. The proposed architecture considers graphs as sets of edges, leveraging an encoder that interleaves masked and self-attention mechanisms to learn effective representations. So this attention-based pooling mechanism or pooling component mimics the functionality of a readout function and is responsible for aggregating the edge-level features into an so where I can't find us where we are here. The masked attention mechanism allows for learning effective edge representations originating from the graph connectivity, and the combination with self-attention layers vertically allows for expanding on this information while having a strong prior. So masking can thus be seen as leveraging specific relational information, and its vertical combination with self-attention as a means to overcome possible misspecification in the input graph. So this is where they get into their edge set approach. For a pair of edges, connectivity translates into having shared nodes between them. We focus primarily on learning through edge sets due to the empirically high performance and refer to our architecture as edge set attention, or ESA. We also show that the overall architecture is effective in propagating information across nodes through dedicated node level benchmarks. The ESA architecture is general purpose in the sense that it relies only on graph structure and possibly node and edge features, and is not restricted to any particular domain. ESA also does not rely on positional, structural, relative, or similar encodings. It does not encode graph structures as tokens or other language-specific concepts or sequence-specific concepts, and it does not require any pre-computations. So this is interesting from what we're doing with embryos, in that you have, on the one hand, there is a lot of position-specific information. There's a lot of structural information that you can learn from, but having an independent representation actually might allow us to understand variation and things that go across species. So, we might want to understand like the common process of gastrulation, and that might help there, as opposed to having a very specific set of spatial relationships. So I don't know. This looks like it might be potentially interesting in that way. So they kind of go through the related work, and they show the GATT and SAN architectures presented schematically. So here is GATT attention, where you have an adjacency matrix. You have a node set that goes into the GATT attention mechanism. That is where you have pairwise relationships. They're concatenated into this matrix. The adjacency matrix is then added to that. You have a linear rectifier, ReLU unit, and then softmax, and then that gives us an output. The GATT model is where we have these GATT attention mechanisms lined up serially. We have the node set that comes into this input is for GATT attention. We have the adjacency matrix is also input, and then we have the node readout, which is after a series of GATT attention mechanisms give an output, we get the node readout. For the SAN attention model, we have the adjacency matrix and then a graph complement. The adjacency matrix feeds into a mask dot product attention, and so does the graph complement. And then you have this term here, and then we have the inverse, and then we have the residual. It's calculated here, and then we have MLP, and then output there. So the SAN model is where we have SAN attention arrayed in serial. We have the node set, the adjacency matrix, and then the node readout. Okay, so that's the difference between those two methods. Okay. Then I'm just looking through the paper to see if there's any kind of what they're actually doing with this. Okay, so the molecular learning here is as learning on molecules has emerged of one of the most successful applications of graph learning, we present an in-depth evaluation that includes quantum mechanics, molecular docking, and various physical chemistry and biophysics benchmarks, as well as an exploration of learning on 3D atomic systems, transfer learning, and learning on large molecules of therapeutic relevance or peptides in general. This is a specific, I guess, data set for QM9, and this just kind of shows this results here, this table, and that doesn't really capture the imagination that much, but it is one of their tests. And then they have docstring, which is this data set where it consists of molecular docking scores for over 250,000 small molecules and 5 high-quality targets. And so they use that as a benchmark as well. They have MoleculeNet and NCI, so this is a series of benchmarks from MoleculeNet, as well as 2 benchmarks from NCI, National Cancer Institute, consisting of compound screen for anti-cancer activity. And they also compares with other graph methods like GraphFormer and Token GT, and those perform poorly. So this performs on small sample sizes, relatively small sample sizes. And then there's PCQM for MV2, which is a quantum chemistry benchmark, and then it performs on that. And then the Zinc data set. And then this is a benchmark where a number of competing methods actually performed well, and kind of shows what the results are here. And so this just shows that ESA can match a lot of the state-of-the-art models without structural encodings and significantly improves upon the result when augmented. So it just shows that structural encoding information isn't always necessary, depending on how the data set is structured. So it's important. So this is just kind of interesting from the perspective of, given the input data set, what is the performance of this method, and what are the strengths and weaknesses? So I'm not really sure, how our embryo data would fit into this, but it does give us some guide to maybe what that might look like. Okay. Then they have mixed graph level tasks where they examine a suite of graph level benchmarks in various domains, including computer vision, bioinformatics, synthetic graphs, and social graphs. ESA generally outperforms all baselines. In the context of state-of-the-art models from online leaderboards, ESA already matches or competes with the best models on well-known datasets, such as MNIST, MelNet Tiny, and CFAR10, without using structural or positional encodings. We also evaluated XFormer and Polynormer on a representative selection of datasets, but the two baselines were uncompetitive with ESA. Then they use node-level benchmarks, and this is where they built a prototype with an edge-to-node pooling module, which would allow ESA to learn node embeddings with good results. However, this approach does not scale to the millions of edges that some heterophilous graphs have. For these reasons, we revert to the simpler node set attention formulation, or NSA formulation. NSA performs well on all 11 node level benchmarks, including homophilous, heterophilous, and shortest path tasks. We have adapted GraphFormer and Token GT for node classification, as this functionality is not originally available, although they require integer node and edge features, restricting their use on some datasets. Okay, so they go through that. And then they talk about the effects of varying the layer order and type. These are kind of details in terms of how you set up the representation. And so that's here. And then they talk about enhancing ESA for state-of-the-art performance. So ESA has a strong foundation for graph learning and has not leveraged positional and structural encodings or other helper mechanisms for the most part. These models were already outperforming or matching the state-of-the-art on several datasets. To explore the potential and compatibility of ESA with additional transformers and components, we further tune hyperparameters such as the optimizer and explore the utility of structural encodings and/or problem-specific modifications for input graphs. For clarity, we refer to this extension as ESA Plus and report our results in Table 6. The results of all 6 datasets are state-of-the-art and top of the online leaderboards at the time of this writing. Okay, then they have things like time and memory scaling. They show some graphs here. And then they talk about the number of parameters for all methods and their configurations. ESA has a fairly low number of parameters. GAT and GAT version 2 have rapidly increasing parameter counts, likely due to the concatenation that occurs between attention heads, while PNA also increases significantly quicker than ESA. Lastly, we demonstrate and discuss one of the bottlenecks in GraphWormer for the dataset HIV. This lack of efficiency is likely caused by the edge features computed by the GraphWormer, which depend quadratically on the number of nodes in the graph and the maximum shortest path in the graph. Okay, and then they also have some things about explainability. And then in the discussion, they kind of summarize what they found. ESA utilizes line graphs where the edges from an input graph are transformed into nodes in the line graph, establishing connections if they share a common node in the original graph. This transformation preserves isomorphisms, meaning that if two graphs are isomorphic, their line graphs are as well. However, the reverse is not necessarily true. Notably, some non-isomorphic graphs that are indistinguishable by the 1WL test we talked about before becomes distinguishable through their line graph embeddings, possibly highlighting richer information conveyed by edge tokens as opposed to node tokens in some tasks. Line graphs naturally emphasize the important motifs like triangles, cycles, and cliques, which are critical for tasks such as predicting molecular properties. So this is interesting because when we're talking about topological data analysis, that's the sort of thing you're doing. You're looking at those kind of simplices where you have these shapes, triangles, and other types of cycles that are shaped in a certain way. And then you're dealing with cliques, and those have a certain shape as well. So this is interesting that there's this convergence. Furthermore, line graphs tend to balance vertex degree variance, maintaining a more uniform distribution, which enhances the regularity of message passing. This edge-focused communication strategy aligns with previous explorations in GNNs, such as in ChemProp, which hypothesized improved accuracy resulting from handling information propagation more effective than node-centric message passing. So this is, of course, another advantage here, or thing that we can explore, is message passing between different parts of the network. So usually if we're talking about an embryo, we might have a network that grows, or we might have signaling that happens between cells, or we have cell divisions, which are basically where you generate two nodes from one. So they split and they move across the graph topology. So this is all kind of, a mechanism we'd like to capture. The message passing, of course, is a method we could use, but then, of course, it's not really message passing. So we'd like to have maybe a different type of method where we can look at how the vertices change over time. So this is kind of interesting. We're trying to map this problem to what they're doing in graph neural networks is the state of the art. And it's not easy. Sometimes you have to sort of contort the data. And I mean that in a good way, not in a bad way. So that's kind of a nice observation on their part to, kind of suggest a path forward for us. So the takeaway from our extensive study is that the proposed approach is well suited to be a simple yet extremely effective starting point for learning on graphs. Indeed, preliminary results on enhancing ESA with positional encodings indicate state-of-the-art results on several datasets. Moreover, the approach is favorable computational complexity and scales better than strong GNNs and some graph transformers. ESA also does well in transfer learning settings, possibly paving the way for more research on foundational models for structured data, and in drug discovery in particular, where the problem arises frequently in property prediction for expensive high-fidelity experiments. Okay. So then they have their methods at the end. Yeah, that's a great paper. Thank you for pulling that up, and hopefully we can think about that a little bit more and come up with a good sort of application there. I think that would contribute to the stuff we're trying to do with graph neural networks and developmental graphs and things like that. All right, did we have any more comments or questions on that paper? Shreyas? Yeah, go ahead. All right. Okay. Yes. All right. So I want. What was that paper you showed? The paper that you're basing this on. Yeah. All right. That is Ed Boyden and Aviamini. Yeah, Max Tegmark. Yeah, this I think is like their work. We were talking about this on Saturday with. I was talking about this with Morgan. This is the atlas they're building of C. elegans. Actually, it's towards a more accurate atlas. So they're trying to use things like expansion microscopy and uh, optogenetics to probe cells and things and get a better measure set of measurements for where the neurons are in space. So the coordinates we have are from cell tracking experiments, and you just take a bunch of worms, you put them under a microscope, you have images in a movie, you do some uh, image processing, you basically segment the cells and their nuclei or their centroids, not really the nuclei necessarily, but and then you track the positions over time. And then you do this for a bunch of worms and you get a data set that you can sort of average out and find like the position. And what they're doing is they're trying to getting at a finer grain scale. They're using some more advanced techniques and other things like that. So that's what they're trying to do there. And then, we talked in Saturday about the whole brain emulation stuff or whole organism emulation stuff that some groups are trying to do, this group in particular. And so that means that you want to build like a model of the entire connectome or entire organism and then make it functional and then simulate all of that. So that's kind of why they're putting this method together. I've not seen this exact paper. Is this what journal? BMC Bioinformatics. So this is what go down a little bit. If you have the abstract, yeah. So they're looking at using aligning and matching tags, mapping that to an atlas, and they are trying to improve on the accuracy of the atlas itself. And they're using a lot of techniques, also like uh, DNA barcoding and things like that, to get really good representations of the cells. So you have the volume of the cells represented and the what the cells are and what their molecular contents are. So yeah, this is good. And then so you're showing you're trying to use this in your work, and you were showing that you're also using attention mechanisms in your code. So that was kind of interesting that um, yeah, yeah. So what about the attention mechanisms? Like, are you doing anything interesting with those or? Yeah, yeah, that'd be good. Yeah. Right. Well, that's not great. That's great. Now, the feed forward bias, what do they mean by that? Just to refresh your memory. Yeah. So yeah, I think what they mean is like in development, you get kind of these connections between two neurons, and it might be like random and bidirectional, and then you need to have some sort of sensory motor coordination. So you need to go from the sensory neurons to the motor neurons and make sure that that's like one direction, and so you can have like a coordinated system where you can have feedback, but it's not in the same set of connections. So that sorting happens in larval development, and that's what they're getting at with feed forward bias. Like basically, that this connection is going to be feed forward because it needs to be for the function to happen, unfold. Yeah, yeah, yeah. That's great. Well, thank you. Yeah, that was good. Yeah, yeah, that's good. That'd be good. Thank you. Thank you. All right, so I wanted to look at Susan posted. This is the Micheletti paper. This is basic tensegrity concepts and calculation methods. So I'm going to go over this paper next. All right, so this is basic tensegrity concepts and calculation methods. It's a book chapter in tensegrity systems. This is kind of going over. So she said that Susan said that the math, it's a nice basic tour of the mathematics. And then I have something to show that I generated from NotebookLM about like from the papers that we were doing in the presentation I gave several weeks ago, and what it says about the different mathematical approaches. So we can go back to that later. So in this chapter, the fundamental notions about tensegrity systems are presented together with their basic calculation methods. After giving a general introduction to the main tensegrity concepts, we dive into the analytical treatment of the statics and dynamics of tensegrity systems. So remember, we have the statics and dynamics. They're separate things. So you model something as sort of this moment in time versus something that's changing over time. And then in particular, we introduce the equilibrium and kinematic compatibility equations. So that those are, of course, we need some sort of equilibrium equation and then some sort of kinematic, component to it to characterize the motion. And then describe the four structural types to which tensegrity systems may belong, characterized by the presence or absence of self-stress states and internal mechanisms. So they propose their four structural types of tensegrities, and that's something we can talk about later. We then pass to quasi-static processes and give the notions of pre-stress stability and superstability. So we look at a quasi-static process. We kind of formulate something about pre-stress stability and superstability. And then, of course, these both depend on the properties of geometric stiffness and tangent stiffness operators. We need two operators, one for geometric stiffness, one for tangent stiffness. Notable exceptions are then reviewed to illustrate nonlinear behavior of tensegrity systems. Dynamic processes are described next by deriving the motion equation. So we need to have some sort of nonlinear large displacement process, we also need to have a linearized form for small amplitude motions about a certain equilibrium configuration. So we start with an equilibrium state. We then add in these small amplitude motions, which are just small perturbations or they're small movements in response to some load. And then you have the large displacement processes, which change the shape considerably or change the rotation. And we need both of those components. The last part of the chapter introduces form-finding methods and presents in detail three of these with simple implementation, an analytical method for symmetric systems, the virtual cocoon method, and a marching algorithm to change the shape of a tensegrity system by controlling edge lengths. So remember we talked about the different form-finding methods before, and we talked about how that can be computationally heavy in terms of having to go through a very large conformational space and search for valid tensegrities. And so this is, again, they're going through these different analytical methods. And I don't know what the sort of degree to which they're hard to compute or easy to compute, but they go through these. Then the latter method is applied to the case of a deployable ring for space antennas. So that's not biological, but it's still a good way to kind of show how form finding works. We talked about form finding in the context of soft robotics, and that's, of course, where you have robots, and this is analogous to an embryo because it's a sort of a functional system that, behaves, and how those, how they did form finding there. It was like sorting through a bunch of geometric configurations. In this case, they're doing it in different ways, where they kind of use an algorithm to change the shape in a certain way, and then they are able to apply that to some mechanical system. So good. I think so basically the three mathematical sort of components here is that you have sort of these definitions of different global states of the system. You have these operators that map to that, so you have to have ways in which you can describe mathematically that global state. Then you have these motion equations. You have these two different components of motion, nonlinear and linear. And then you have these form finding methods, which are selection methods from a conformational space, basically saying, what is a valid tensegrity? What isn't a valid tensegrity? And so this is a nice article. Now I took the readings that we had from the presentations that we did a couple weeks ago, and kind of going through the basics of how you would get, what are the mathematics that are involved in understanding tensegrity structures. So I just kind of pulled these up. So I don't have this. I haven't fully cited these. I have to go back to the notebook and fully cite them. It's just citations from the papers that were in that presentation, and I just had them in the folder. So that's how NotebookLM will just cite from those papers and cite specific things to tell you kind of why it's telling you this. But I mean, this all looks pretty consistent. So I queried the model. What are the major approaches to mathematically modeling tensegrity? What types of math are used? So it says mathematical modeling of tensegrity structures primarily focuses on two key problems. The first is finding self-equilibrated configurations, which is what it terms form finding, and then guaranteeing stability. So you need the stability guarantee on top of the form finding. These approaches rely on a diverse range of mathematical fields, including linear algebra, calculus, graph theory, group theory, and rigidity theory. So this is sort of thinking about the fields of math we need. I would say, of course, matrix algebra, which is kind of guess linear algebra, but and then some other dynamical systems concepts as well. So that's maybe where I would add in some things. And then, of course, graph theory, which we've been talking about with respect to embryo networks, but also I think graph theory in terms of the basic relationships between edges and nodes. Okay, so then major approaches to modeling tensegrity. So the first is form finding or shape finding. This is the process of determining the configuration and pre-stresses of a structure in a state of self-equilibrium. So because geometry and pre-stresses are highly independent, we use three methods. For this, we use numerical methods, we use analytical methods, and optimization methods. So we talked about optimization as sort of finding the best structure for this. We also talked about analytical methods, which can derive an exact solution. And you can, if you have a high level of symmetry in your structure, that's a very good method. And then numerical methods, which are, of course, allow you to work with like force density and dynamic relaxation and static nonlinear structural analysis to kind of come up with sort of the structures that you think are stable and super stable. Then you have stability analysis, which is where you can determine if a structure can return to its initial configuration after a small disturbance. We use three criterion to determine this. We have stability, which is where there's a locally minimum total potential energy. So it's like if it returns to that minimum of potential energy, then it's stable. If it does not, then it's unstable. There's pre-stress stability, which is examining stability assuming infinite axial stiffness. And then superstability, which is the most robust criterion, where structure is always stable regardless of material properties or pre-stress level. So you see that there's this idea of stability, which is the weak case, and then superstability, which is the strong case, or the strongest case, where you have sort of the most exclusive case of, that supersedes material property dependence or a level of pre-stress. So we can get from very general answers to very specific answers. Then there's topology identification, and this has to do with like topological data analysis and network topology and things like that. So I mean, they're what I'm saying is that this leads us to other areas that we're interested in, and can tell us things about, say, like an embryo network. This involves determining a proper connectivity between nodes that allows for a self-standing structure. Newer methods utilize abstract graphs, while fundamental tensegrities with larger, complex superstable structures. So basically, you're using these kind of you're using abstract graphs and then kind of mapping that to the tensegrity structure. The abstract graphs, then we use those to kind of find the optimal configurations, and we map that out to the tensegrity structure. It also is interesting because then we can see, first of all, if we have some interesting property of an embryo network, can we replicate that in a tensegrity structure? In other words, if we have like a change in shape that we saw with gastrulation, can those changes in shape, if we're characterizing them as they're kind of forming, do they relate to tensegrities and the transformation of a tensegrity structure? And maybe that tells us something about how the way in which those shape changes happen, how the structure is actually maintaining stability. It also tells us something about like the simplices that we talked about earlier, or the topological features in a graph. So sometimes you have triangles, sometimes you have diamonds, sometimes you have other structures that are important for sort of, being these core primitives of stability or core primitives of, say, signaling or something. And those can be, we can map those to the valid tensegrity structures. So not only does a tensegrity structure need to be super stable, but it also needs to have sort of the properties of a biological graph that's functional. And so then it says types of mathematics used, of course, linear algebra. And this is important for modeling equilibrium, defining topology, having an equilibrium matrix to relate forces to external loads, and then force density matrices to transform nonlinear equations into linear ones. We can also use eigenvalue analysis, which is a subset of linear algebra. Then we can use calculus. We need to use calculus to look at the first order partial derivatives that allow us to find stationary points for equilibrium. And then we can use second order derivatives such as Hessians or tangent stiffness matrices to determine stability. We can, of course, use graph theory as we just described. We also can use group theory, which allows us to look at symmetries and then different equivalent symmetries. Rigidity theory, which allows us to sort of uniquely define stability, rigidity, and pre-stress stability. So those are all kind of important. Kinematics, of course, we need to understand movement and transfer of motion and things like that. And that's where we have especially like these open kinematic chains or closed kinematic chains. This is the closed kinematic chain reference here, where multiple bars are coupled into mechanical loops. So in kinematics, there's a force generated at some point, and it gets propagated out in a series of components or chains. And that, if those components move, the motion component becomes amplified. If it's a closed kinematic chain, then those motion differences self-amplify the structure. And so we can actually look at those kinematic chains, whether they're open or closed, and kind of predict what the dynamics will look like and how more motion and force is transferred through the entire system. And then I asked it about the superstability hypothesis in particular, and this has it kind of analyzes this hypothesis. This is a hypothesis that we've talked about in the past, which is basically the idea that in biology, superstability or tensegrities are really important, and that they are important for homeostasis and maintaining the structural integrity of organisms. So the core principles of the hypothesis, number one is robust stability criterion. So it gives us a good, solid stability criterion. It allows us to look at biological transformations, or how organisms and their shape grow and develop and remodel without losing stability. 3 is inherent internal control. So, this is where we want to maintain homeostasis. We want to maintain stability, not just structurally, but functionally as well. And then it says, unlike traditional biomechanical models that view the body as an unstable machine requiring catch up stability from the nervous system, the superstability hypothesis argues that stability and motion are controlled from within the structure itself. This allows tissues to respond instantly to physical forces, bypassing the dangers of transient muscular latencies. So what they're saying is that like this kind of approach allows us to merge more effectively what's going on in the phenotype with what's going on in the connectome. And a lot of times when we model these things, we're modeling them sort of as separate entities, and they're not connected. They're not integrated functionally. So then they talk about the significance across scales. So molecular level, cellular level, whole organism level. These are all kind of components of those super stable tensegrities. Okay, so that's all I wanted to talk about there. I wanted to get a little bit into some of these things. Shreyas had a question. So go ahead, Shreyas. Hi. Yeah, so some like with group theory, what it does is you have a bunch of nodes, and those nodes are connected in a certain way. You can describe like connectivity, but sometimes there are different things like translation and rotation that are important as well. So you know your your nodes, that like you can describe a structure statically, and then it'll rotate. So sometimes you're interested in the rotation of the phenotype. The graph is maybe stable in one configuration, but if you rotate it 90 degrees, it's not stable. What you want to do is you want to find those stable modes. Go ahead. Yeah. Yeah, I think so. I mean, that might be a little bit more than we need, but like that would be interesting. Like to know kind of what nodes in the embryo are invariant. In other words, if they get rotated, then they are not, it's still a stable structure. This is actually important in evolution, where you have different transformations across organisms. You have the same underlying genes and developmental process, but it's shifted in ways that are changed. The way in which things are configured. It might change the orientation of the cells, or things might get flipped around, and they still remain stable, at least as far as the ones that survived. So that's interesting there. But I think yeah, these basic transformations are interesting in development because you do get a lot of transformations of different structures that flip around, and you start with like things at the anterior end and they migrate inward, or things kind of fold, or things like that, and it's important to know kind of how that's all being kind of coordinated, not just like in terms of molecular guidance, but like the structural aspects of that. Yeah, 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 no problem. Okay, so do we have any other questions? Susan, did you have any comments? Oh, Barshan, did you like to speak? Yeah. Yeah. All right. Susan, did you have something to say or which one? Oh, I got that. Like all of the papers that were, I think some of them you sent me, some of the ones I looked up. I put those into NotebookLM, which will basically get things out of every paper and kind of synthesize them. So it was all the papers, and I can prepare I have the citations in the notebook. I just need to map them over to the Word document, and that would tell like exactly where it's coming from. Yeah, yeah, yeah. All right, it's good. Yeah, yeah, yeah. Well, yeah, it's always useful, I think. All right, all right. Well, yeah, I think that's all for today. Thank you to our presenters, Shreyas and Barshan, and keep up the good work. And Varshith, I'll look forward to seeing the draft paper. And Susan, good luck with the tensegrities.